All right, everyone, this is a flipped instruction over the plasma membrane. I am expecting that you are taking notes, so you will want to take out your ISNs and take notes and follow along with this video. The plasma membrane, it's a flexible boundary that kind of separates a cell from its surroundings. This image shows fantastic kind of layers of the biphospholipid focus, and I'll get into that in a little bit. But basically, what the plasma membrane does is it regulates what actually comes into the cell, and it provides protection and support. And by regulating what comes into the cell, this is called selective permeability. Selective permeability means that basically some molecules are allowed to pass through the membrane at any time. Other ones are only allowed at certain times or in limited quantities or amounts. And then some molecules are never allowed through. So, like I mentioned a second ago, phospholipid bilayers are basically a double layer sheet that makes up nearly all of that plasma membrane. And phospholipids are made of glycerol, two fatty acids, and a phosphate group. So if we look at this image right here, kind of focused in a little bit further, you can see there's like a headpiece and two tails. There's hydrophobic and hydrophilic focuses of this membrane. Notice how that head with the tail and the head and the tail, they kind of face each other there. This is another kind of imagery of that. So here we have that head, which is the phosphate, and then the tail, if you will, is focused of fatty acid chains. And so they form together to kind of allow certain things to come in, like carbohydrates, or it allows proteins and whatnot to go through that layer. Plasma membranes contain protein molecules that are embedded in that phospholipid bilayer, and carbohydrate molecules are attached to many of the proteins. The organization of all these structures is called the fluid mosaic model. And that is kind of all that I'm going to have you focus in on. Once again, spend a little bit of time focused on the boundaries themselves. I want you to know the different parts of that little plasma membrane. It's going to be important for the lab that we are going to do in a little bit.